Good afternoon, everybody. Thank you for being on time. We will get started. So first of all, I would like to say the quiz was relatively well done in general. The class average was quite good. And hopefully you feel that the quiz accurately reflects what was being taught in the class and that I've helped you prepare for the quiz adequately. And if you have questions or concerns, or if you need extra help, as always, please feel free to contact me. Now, because this is an online course, our department have decided that we would not be sharing the results of the quiz. We won't be showing you which question you got wrong, because we want to maintain the integrity of the test bank of the test questions. I understand that is a little bit disappointing for some of you, but as I said, that is just a constraint of how the course is being delivered rather. So let's get started. Today's lesson is on cell, and it's a quite easy lesson. It shouldn't take very long. So first one. State the 4 principles of the cell theory. Now, cell theory is one of those kind of groundbreaking theory that has led to many other breakthrough in science. By establishing the cell theory, people understand that there are actually things beyond what our naked eye can see. Right? There are all these cellular things that are happening, and that helps us understand things like bacteria, things like viruses. Eventually, those are, of course, disease-causing agents. So, by having a cell theory, it kind of laid the groundwork for many of the cellular biology that comes afterwards. So, say the 4 principles of the cell theory. Well, the first part of the cell theory, or it's not really in any particular order, but like one of the 4 parts of the cell theory is that every organism consists of one or more cells. Okay, so if you think about a bacterium, right? Plural is bacteria, right? So 1 is bacterium. That is equal to 1 cell. Okay. And if you think about a cat, right, then it has like many cells. Like humans, we have like trillions of cells, right? So every organism, if it's alive, organism implies it's alive, right? So it's going to consist of 1 cell or more cells. Okay. What is a cell? That's the second part of the cell theory. A cell is the smallest unit of life. Okay, so it exhibits all the characteristics of life, and we learn about the characteristics of life in the first week, right? There are complexity to it. It requires energy, is able to maintain homeostasis, right? So a cell is the smallest unit that exhibits all those properties. Okay, and to add to that, it is individually alive, okay, as part of a multicellular organism. I always forget to change these worksheets to make it more space, and especially this one. I just taught a class earlier today, and I realized it's really tight in space. So I do apologize for that. If you need to have an extra piece of paper, please go grab one. You might need that. Okay, I'll try to remember for next time to add more space to it. So another point of the cell theory is that all cells come from the division of pre-existing. I'm trying to move this out of the way. Okay, individually alive. Okay, pre-existing cells. Okay, so we actually have a name for that. What do we call the division of cells? Anybody remember? You know, if you took high school biology, or if you watch the lecture, or if you just hear it from the internet or something. So Christian said mitosis. That is right on. Mitosis, cellular division. Okay, so nobody knows where the first cell came from. Okay, like if you believe in evolution, then maybe by chance, all these things come together and form a cell. If you believe in creationism, then you have your own theory. It doesn't really matter. Nobody knows for sure. Okay, you can choose to believe what you believe in. But what we do know now is that all the cells must come from the division of a pre-existing cell. Okay, so that new skin cell that you got, right? That is trying to heal your wound, right? It came from the division of a pre-existing skin cell. Okay, so that's part of the cell theory. And finally, the cell theory states that cells contain hereditary material. Hereditary materials that can be passed on during mitosis. So what kind of hereditary material are we talking about? Well, it can be DNA or sometimes RNA. Okay, so these are the 4 principles of the cell theory. Are we good? All right. Next, fill in the blanks with terms that correctly describe the organization of life from smallest to largest. So last class, we talked about atoms. Atoms are basically the smallest unit of an element, right? And elements are all these things that you find on the periodic table. So, oxygen, carbon, nitrogen, phosphorus, hydrogen. These are the common atoms that makes up an organism, right? That makes up humans, at least. And so the atoms can combine together to form molecules. Okay, so molecules are like, small things like water. We definitely need that. We need maybe glucose, right? Not maybe, like we definitely need glucose as well to burn as energy, right? But these are small molecules, okay? Last time we learned about the macromolecules, large ones, really big, thousands of atoms as opposed to just 3 or 24, right? And of course, we have things like carbohydrates, right? Lipids, proteins, and nucleic acids. Those are macromolecules, right? Okay, what is next? Well, the macromolecules can actually, form various structures as well. And in relation to what we're learning today, the next level up would be these organelles. Okay, so can someone tell me what is an example of an organelle that you remember from lecture or wherever? Give me some organelles here, people. Anybody know any organelle? Thank you. So Megan said nucleus. That's excellent. Jyoti, forgive me if I'm pronouncing that wrong. Ribosome, that's another one. Okay, so I'm gonna put ribosome, right? Nucleus, and a bunch of other ones that we've learned, right, in the lecture. Mitochondria, all that stuff. Okay, so these organelles are like mini organs that are functioning within the cell. And so if you move up one more level, then it would be the cell. And it could be any cell. It could be like a heart cell, we can say, a heart cell. Could be a muscle cell, could be a liver cell, skin cell, brain cell, any cell. Okay, so sometimes you would have a collection of different cells that would then form tissue. Okay, so we have many tissues in the body. We can have like connective tissue, for example. Connective tissue. Okay, there are actually different types of connective tissues. You have your fat tissue, that's a type of connective tissue. You have your fibrous connective tissue that forms things like tendons, right? So many different connective tissues. And as the name implies, connective tissues help you connect stuff together, right? Okay. So different types of tissue will form an organ. So an organ would be something like a heart. And then many organs will form an organ system. And can someone tell me what organ system does the heart belong to? Like, for example, like an organ system would be like the, oh, okay, Daniel already answered the question. Excellent. That would be the circulatory system. That's right. Circulatory system. Okay. Yeah, you spell it just fine, I think. Yeah. Very good. Okay, so that's the hierarchy. Make sure you know how to organize it. Okay, I'm pretty sure I put one of these questions on the test, right? Or next quiz, which is going to happen in 2 weeks, by the way. I will remind you on eCentennial as well as during the tutorial. Some of you actually missed the quiz because you got the wrong day. So just a reminder, there is a weekly schedule on eCentennial. Okay, and it tells you when everything is going to happen. And also, I will remind you relentlessly, right? Like I send out email, I post announcement on eCentennial. So just keep on checking those things. And did you know that you can forward email from your eCentennial email to your private email? Like some of you use Gmail, Yahoo, whatever, right? You can actually redirect your school email to those other email servers that you use. Okay, that way you will not miss anything. If I send you an email, it's probably important. Please read it. Do not ghost me. Moving on. List 3 common structures found in all cells. Now, there are so many different cells, right? I talked about there's bacterial cells. There is, human cells, and even within humans, we have the heart cell, brain cells, right? You have plant cells. They are all quite different in their own ways. Okay, they look different, they function differently, but there are 3 common structures that are found in all the cells. Okay, so the 3 common structures are, number 1, they all have some kind of membrane protecting them. Okay, so we call that the cell membrane. Okay, sometimes it's also called a plasma membrane. Okay. And other thing that is common is that there is a cytoplasm. And so if you think of like a cell, right? All the gooey stuff inside, right? All the stuff inside the cell, the gel-like substance that's inside, that's the cytoplasm. Okay, cytoplasm. If you take a Ziploc bag, fill it up with, I want to say water, but it's not actually water. It's more like gel-like substance, like hair gel, if you would. Okay, so you put that in, and then that would be your cytoplasm. And the last one. Does anybody remember what the last one is? Help me out here. Okay, Daniel said nucleus. Now, is that something that will be found in all cells or just some cells? We're looking for something that's universal. All the cells. Anyone else want to say anything? Good try, though. Anyone else? Okay, that's okay. They will. Okay, so Micaiah said ribosomes, and then Fantone said genes, and then we have a bunch of other ones. Yeah, so genetic materials, that's the correct answer. Okay, genetic material. Remember, right? Think about the cell theory, right? Cell theory states that all cells, actually it's the last one. The answer is right here, right? Cells contain hereditary material, right? So that's a genetic material that can be passed on, okay? If there's no genetic material, then it's not alive, okay? Everything that's alive is going to have the genetic material, okay? So those are the three common structures, or maybe structure is not the best word, right? Three common things, characteristics that are found in all cells. Okay, they have cell membrane, they have cytoplasm, and they have genetic material. Now, there are two kinds of cells in general that we categorize cells in. We have the prokaryotes, or prokaryotic cells, right? Also known as prokaryotes, right? Prokaryote. Okay, prokaryotes or prokaryotic cells. These are more primitive types of cells, and again, in the theory of evolution, prokaryotic cells would have evolved before the eukaryotic cells. These are the more complicated ones. So comparing the two, in terms of size, the prokaryotes are smaller, a lot smaller compared to the larger eukaryotic cells. Okay, so eukaryotic cells is so big compared to the prokaryotes that sometimes the prokaryotes can go inside the eukaryotic cells and kind of live inside them, cause all kind of havoc, and that's not a good thing, of course, for the host. Structure. Okay, so the structure found in prokaryotic cells are quite simple, whereas for the eukaryotic cells it is complex. Location of genetic material. They are just kind of, hanging around in some central region, central area inside the cell. Inside the cell. They are not compartmentalized. Okay, that means they're not like in a separate area, just kind of hanging around in the middle. So if you think about like a bacterium, sorry, there is bacterium, singular, right? Then the genetic material would just be hanging around in the middle. Okay, I mean, bacteria sometimes they have these circular DNA as well that are floating around. We learned that in lecture 8, I think, or 8 or 9. I don't exactly remember, but we will. Okay, but the genetic material is just kind of hanging there. It's not, confined in a single place like the eukaryotic cells, right? The genetic material for the eukaryotic cells, they are inside the nucleus. Okay, so no nucleus for the prokaryotic cells, but eukaryotic cell. If this is a eukaryotic cell, you would have a separate circular structure on the inside, and that is where you will find the genetic material. Okay, genetic material. Quick question, just want to see if you guys are with me. Are we humans? Are we eukaryotes or prokaryotes? What I mean is, do we have eukaryotic cells? Are we made up of eukaryotic cells, or are we made up of prokaryotic cells? What are you guys saying? Okay, yeah, I got some response on the chat. Very good. We are eukaryotic cells, but here is the catch, okay? Even though we are made up of eukaryotic cells, and eukaryotic cells are supposed to have nucleus, actually, right? Some of our cells have no nucleus. Did you do that? Anybody have an example of a cell that does not have nucleus? And you can just like, brainstorm, spitball a little bit. Doesn't matter. It could be a guess. I just want to hear from people. I just want to make sure that you're there, that you are engaging. Sex cells actually they do have nucleus, believe it or not. And you know the egg's gonna have like 23 chromosomes, and the sperm's gonna have 23 chromosomes, and then when they come together, baby happens, right? So they do have nucleus. Good guess though. Anyone else? The cells that make up your skin. I mean, skin are dead cells. So before they become dead, they would have a nucleus. I mean, like the skin cell actually like these cells, like it sheds or like dandruff, right? Those are dead, right? But before they become dead, they would have the nucleus. And so Gita said red blood cells, very good. Zafi Raza also said blood cells, very good. So RBC, red blood cells. Okay, that's what RBC stands for. Not the Royal Bank, but red blood cells. They don't have nucleus. It's very interesting. We will learn why next semester. Right? If you end up in my class again, I will tell you all about it. And then platelets, that's another one that does not have nucleus. Okay, so there are exceptions. Like in science, is not everything is like this or that. Okay, there is a lot of exceptions, and we just have to assume that sometimes. Okay, accept that. I mean, not assume. Number of organelles. So for prokaryotes, few. Okay, just a few basic ones to keep it going. But don't underestimate the few organelles, right? Don't think just because there are fewer organelles, then they're, you know, not as sophisticated as eukaryotic cells, because after all, you know, the bacteria can kill us, right? So even though they're very simple, they can still pack a punch. For us, many. And examples for the prokaryotic cells would be bacteria, protists, some protists. Actually, no, just bacteria, not protists. I'm confusing myself here. So bacteria. Anybody know any example of bacteria? I'll give you one that, you know, most of you have probably heard of, like E. coli, right? Coli can, you have some harmless E. coli in your guts, but sometimes if you drink contaminated water with E. coli, you can end up having diarrhea and stuff like that. Any other bacteria that people know of? Anyone that you've heard on the news? Or Megan said paramecium. Okay, paramecium, it's a good name, but that is actually protist. So not a bacteria. Protists are like these things that lives in the pond, right? Like amoeba, right? Those kind of things. Rubella is a virus, but good try. Salmonella is a good one, Daniel. Salmonella. Okay, very good. Okay. There are other ones, like Staphylococcus, right? You heard of people getting strep throat, right? That's Staphylococcus, right? Listeria is another one that you might have heard of. Like sometimes they recall cold cuts and whatnot, right? There's Listeria contamination, right? It causes listeriosis. You know, many more. It's not important. You don't have to memorize them. I'm just trying to make it relevant to you. Continuing, the eukaryotic cells, examples would be like plant cells, plant and animal cells. Animal cells, right? And the paramecium that Megan mentioned earlier, that would be an example of a eukaryotic cells as well. So they belong to a group of organisms called the protists. Right? Do you guys know that there are like 7 kingdoms, right? 5 kingdoms rather of like organisms, right? There are the plants, there are the animals, there are the protists, and then there are these fungus, as well as the Monera, we call them. The Monera is where the bacteria are, and these bacteria that lives in like super extreme environment, right? Like living in like hot springs and volcano stuff. So those are the Monera group. So those are the 5 kingdoms, and they are kind of separate. Okay, and all those things except for the bacteria would be like even the fungus, that would be the eukaryotic cells. Okay, sorry, I move on a tangent here. Let's come back to what we are talking about. But first, let's do a quick poll and see if you guys are still with me. Okay. Am I sharing my screen? Nope, I'm only sharing part of the screen. So here we go. This is a poll question, and I will launch the poll for you to do. Go ahead. Okay, I will end the poll. And all of you got it right. That is very good. So I don't have to explain why. Let's move on then. Back to here. Label the different parts of a mitochondrion. Okay, so singular is R-I-O-N, plural is R-I-A. So this is a mitochondrion. And we will fill this in later, but mitochondria basically is the powerhouse, right? They make ATP, which is the usable form of energy in your cell, in the body, okay? So the mitochondria has a outer membrane, which is outlined here in purple. It's so funny that in all textbooks, they choose to make the mitochondria orange. But as you know, like in real life, if you look at a cell under the microscope, everything is like transparent, right? Because they're so thin, so there's actually no color to it. But I don't know, someone sometime in the past decided that, you know, they're going to be orange. And so it's always orange since then. So here, that is the outer membrane. Outer membrane. Okay. And I know it's not easy to see, but this one here is pointing at these projections, these foldings, if you would. Okay. So on the next quiz, right? Be prepared to label some of these diagrams. It's still multiple choice, but like I would be pointing to a part, and then I would ask you like, you know, what am I pointing at? Things like that. Okay, so this is the inner membrane, and the inner membrane folds into these finger-like projections. That's what they're usually called. And the name for those would be the cristae. Okay, crista is singular. If you add an E, that's plural. Okay, so the reason for that is if you fold it up, then you can maximize surface area for reactions, for chemical reactions. Okay, after all, to make ATP, it actually involves a lot of reactions. And, you know, the more surface area you have, the more reactions can take place at a given time. So it speeds up your ATP production. Okay. Now, the space between the outer membrane, the purple, and the inner membrane is called the intermembrane space. Intermembrane space. And there is a lot of things going on in the intermembrane space, as well as the matrix, which is, you know, the fluid inside the inner membrane. So exactly what happens in all these places and how this ATP being produced, well, we are going to find out in lecture number 5. And that's not going to happen until like after the reading week. Okay, and I'll tell you when that is if you're not sure. But that's the mitochondria. Any question? All right. So, did you know? Mitochondria. Your mitochondria. Did your father give you your mitochondria, or did the mother give you the mitochondria, or both? Let's find out. Let's do a poll and see. Okay. If you think it's all from mom, please choose A. If you think it's all from dad, please choose B. If you think it's both, like obviously from both, then you choose C. Okay. What do you think? Who gave you your mitochondria? It's like, I've never really thought about it until now. Who gave me my mitochondria? Okay, let's anyone else want to chime in? Yeah, okay, that's a pretty decent response rate. Okay, good. All right, looks like we are undecided as a class. I love this. I love it when there are so many different opinions. Well, the correct answer is actually mom. Okay, all your mitochondria came from the maternal side, because the egg. This is the egg, right? Okay, with the nucleus and the 23 chromosomes inside, and then it has all the organelles, right? Mitochondria here, and a bunch of other stuff. Okay, and this is the sperm. Okay, it has swum such a long way to find the egg. Okay, by the time it gets to it, like all the mitochondria are kind of very exhausted, so to speak, because it has been producing energy, right, for the cell. And only the genetic material from the sperm goes into the egg to form the baby, right, to form the zygote, right? So the egg has all the mitochondria that will support the development of the fetus. And so all your mitochondria came from the mother, the egg, which is kind of cool, right? Another reason to thank your mom. Now, another interesting thing is that the mitochondria have its own DNA, which encodes for some of its protein. Okay, so in order for the mitochondria to perform the chemical reactions necessary to make the ATP, it needs to have a lot of proteins. Some of the proteins are inside the matrix, some of the proteins are embedded within the inner membrane. All that proteins are going to be encoded by DNA, right? We learned that last class. And at least some of those proteins are encoded by its own mitochondrial DNA. And just like the mitochondria is inherited from the mother, so is those mitochondrial DNA. Now, sometimes if there are mutations in those DNA, then what happens is it could lead to mitochondrial diseases, right? And depending on the severity and which gene is being mutated, it's possible the mitochondria cannot make enough energy and enough ATP to keep up with demand, and it could lead to a bunch of problems, right? Heart problems, diabetes, high blood pressure, to name a few. Okay, so if you're interested, you can definitely look that up. So I hope you learned something interesting from that. Back to our regular program now. So now we are going to talk about the component of the endomembrane system. Endomembrane system. Now that's a complicated word, a phrase, it seems. Oops, I want a highlighter. There we go. The endomembrane system. But basically, endo means within, right? So it is just a series of membrane inside the cell. Okay, and they have related functions. Okay. So first one, right? It's pointing to this part here. That is the rough ER, the rough endoplasmic reticulum. Rough endoplasmic reticulum. Okay, or RER for short. Okay, so let's talk about some description and functions. Okay, so again, like I said, I realize this is not enough space to write everything I'm about to write, but try your best. So I'm gonna cheat a little bit. I'm gonna zoom in, because if I zoom in, I get more space. Okay, and then I'll make this available online afterwards, right? Like I did last time. So I will do the functions in one color and the description in another. So what is this? Well, it is part of the endomembrane system. All of these are, so I'm not going to write that. And what it is it is continuous with the nuclear envelope. Okay, so your nucleus, right, which is what you will find in all eukaryotic cells, it is in the middle of the cell usually, and it's surrounded by a membrane. That membrane is called the nuclear envelope. And that envelope will extend into the cytoplasm, forming the rough ER. It is called the rough ER because when you look at it under the microscope, now you can't just use like any regular microscope, right? Like the one that you've used at school, for example, if you've used a microscope before, that typically is a light microscope. You won't be able to see the endoplasmic reticulum with a light microscope. You need something that's much more powerful. Okay, if you want to take a good look at the ER, you will need something like a electron microscope that gives you very, very high resolution. Okay, so what does it look like? Well, it looks really bumpy under the electron microscope, and that's because it has a lot of ribosomes on them. So they are covered with ribosome. Okay. And what does that do? What do they do? Well, what they do is that they will the ribosomes synthesize proteins directly into the RER. The protein folds up, right, into a secondary, tertiary, and maybe quaternary structure, and pinches off. The rough ER has a vesicle. Okay. So what does that look like? Zoom out a little bit. So we have the, oops, it's too thick. We have the rough ER here. Okay, it's just a bunch of channels. Okay. And then you would have your ribosomes on them. Right. So like this ribosome here would produce a protein, like a polypeptide. It would link all the amino acids together. Okay. And this long chain will fold up. Right? Like if I zoom in over here, if I take this location and zoom in, okay, so maybe it's like this. This is the ribosome, okay, and then you make the protein, and the protein will fold up into a 3D shape, and then it will move towards the edge, and then this part is gonna like bubble off. It just pinches off, and it will have the protein in it. So this here is called a vesicle. Any question about that? It's probably not the most, pretty drawing, but hopefully it gets the point across. Any question? Okay. Next one, we have the smooth ER, smooth endoplasmic reticulum. Okay. So what can we say about that? Smooth ER. Okay. Also part of the endomembrane system. And then it is not covered with ribosome. So appears to be smooth under a microscope. Okay. What does it do? The smooth ER is responsible for synthesis of lipids. Okay. So example, lipid hormones, right? Steroid hormones. We learned that word last time. Steroid hormones. Okay, such as testosterone, progesterone, estrogen. These are steroid hormones, and the smooth ER is responsible for doing that. Okay, so you would expect to have. So you would expect to have lots of smooth ER in what kind of cells? What kind of cells in the body would you expect to have a lot of smooth ER if they're responsible for making steroid hormones such as testosterone, I said, right? Progesterone, right? They all end with ONE, right? That tells you it's a steroid, right? Estrogen. Where do we make those in the body? Anybody? Where did the testosterone come from? Where does the progesterone, estrogen comes from? Anybody know? Really? Nobody knows? The sex organ, the pituitary gland, the brain. Okay. What sex organ? What, like in male? What is that? Or in female? What is that? Sex organ is called gonads, right? GONADS. So what's the gonads in male and female that produces these things? Testicles, that's right. So such as expect to have lots of smooth ER in cells of the testicles. Oh, sorry, can't spell testicle. Yeah, and for female, ovaries. Very good. Okay, testicles and ovaries. Good, good. Now they are also, the smooth ER is also responsible for breakdown of lipids and detoxify drugs, right? So which organ, what organ is responsible for detoxifying drugs? Anybody know? I'm not talking about like bad drugs. Okay, like you take antibiotics, you take an Advil, you take a Tylenol. It's gonna get detoxified in the body. It's gonna get broken down, metabolized, right? Yes, liver, very good. So you would expect to have smooth ER in the liver as well. Okay, in liver cell. Okay, like you would have more of those. I'm not saying you would only have smooth ER in the liver cells, but you would have more smooth ER in liver cells because of the detoxifying drug property. Okay, you will have smooth ER in almost all the cells because there are other metabolic lipids that needs to be broken down, and so other cells would have smooth ER as well. Does that make any sense? Am I explaining it clearly here? Everybody know what I'm talking about? Okay, moving on. Next, we have this thing. Okay, so the ER, like I draw it like this, right? ER is more like a continuous channels of membrane. For the Goji body, it's more like stacks of pancakes. Okay, it's not like one big thing that's interconnected. It's more like stacks of membranes. Okay, so that's the difference. Even though they look very similar, but that's just because they are all part of the endomembrane system, right? So this is the Goji body. Goji body, also known as the Goji apparatus. Okay, depends on what books you read. Like the newer ones, I think they just call it Goji body because it's just easier, right? Like it's already complicated enough. We don't have to give it difficult names. But I'm just telling you, just in case you read about it. Okay, so let's do the description. And description of the Goji body. Again, part of the endomembrane system, and it consists of layers of membranes stacked together. They're not really touching each other, right? They are just in very close proximity. Okay, so this picture is kind of hard to see, so I'm going to draw like a easier one to try to help you understand what exactly it does. Okay, and then I will put it in words for you. Okay, so this is just a simplified Goji body. So that vesicle that contains the protein from where? From rough ER, for example. This is going to fuse with the Goji body. Okay, so the protein will go in to the first layer. Now, at the end of these proteins, there are usually some kind of signal tag, okay, like a few amino acids at the end of it that tells you, they're kind of like, barcodes. And then the Goji body will scan those barcodes and it will tell them what to do with it, okay? So sometimes, right, they will repackage it into another vesicle that will move forward, right? And then the protein would get like modified, like you would add things to it or something, and then you would repackage it and then move it forward. And then sometimes you have to move it backwards and forwards, like it goes back and forth a couple of times, depending on the type of protein. But finally, you will repackage the protein into a new vesicle. Okay, so essentially what the Goji body does is that it will sort, modify. Oh, sorry, this is function. I should use blue. It will sort, modify, and repackage proteins, right? And these are proteins that are from the rough ER. Okay. So there are a couple of destination for these are repackaged vesicles. Okay, so they might go to become a membrane protein. We'll learn more about that next time. They might be released from the cell. Okay, so let's say this is a pancreatic cell, and you just made some insulin, and then you just had a big meal. So then the insulin will move to the cell membrane, and then it will release its content. So the insulin will go into the bloodstream. Right? It's also possible that you did not eat yet, so you don't need the insulin. So instead of releasing from the cell, it will stay in cytoplasm. Okay, and sometimes the protein that are staying in the cytoplasm doesn't have to be in a vesicle. It could just be like a free flowing protein, and still sometime it could become the next one. Okay, and that next one is the jumping all over the place. Next one is the lysosome. Okay. So what is the lysosome? Lysosome it's a vesicle. It's part of the endomembrane system. It's a vesicle that branches off from the Goji body that contains multiple enzymes, right? So maybe it's not just one protein. There could be other proteins coming in, and then the Goji body will be putting them together and put it in one package and then that creates the lysosome. It's like Amazon, right? Like you order 3-4 things. So at the sorting center, like I don't know, I'm just guessing. I've never seen the inside of what they actually do in the warehouse, but I assume they would have to get all these different packages that you order, put in one box, and then ship it out, right? So the lysosome is kind of like that. You get all these enzymes. Enzymes are proteins, right? Remember, right? That are produced, and then you put it in one ball, and then you package it, and that becomes your lysosome. Okay, so it contains multiple enzymes. So what does the lysosome do? Well, a couple of things, right? A couple of things. The lysosome can break down macromolecules. It could break down bacteria, right? Old organelles. Okay. And sometimes if the cell gets too old, then the lysosome can also release its content to kill the cell. It can release all the enzyme to trigger cell death. Okay. Sometimes it's better to kill off one cell that's not working, and unlock some of the resources. So that's what it is. So someone mentioned red blood cells earlier. They don't have a nucleus. What's the other color besides red blood cells? What other color cell do we have? The white blood cells, right? Okay, so you have many different white blood cells, okay? And you will learn all about it next semester. One type of white blood cells, it is called a macrophage. Macrophage. Okay? Macro means big, phage means eater. So this literally means big eater. And if you want to see one of these things eat stuff, just go on the YouTube, internet, search macrophage in action, and then you will have lots of video. There are one video where you can see the macrophage like chasing a bacteria. It's actually kind of comical. You should definitely check it out. So the macrophage would have a lot of lysosome. And these are lysosome. And what happens is if there's a bacteria, then the macrophage will be like Pac-Man, right? You guys know Pac-Man, right? It will eat the red bacteria, and it will surround it in a vesicle like this. And then this vesicle will fuse with the lysosome. Sorry, I keep on saying lysosome, which is something else. Okay, and the lysosome will fuse with this vesicle, and the enzyme inside the lysosome will break down the bacteria. Okay, so that's an example of a cell that has a lot of lysosome. Okay, that's that. Whew, that was a lot of stuff. Anyone have any question? Now, just on a side note, we in lecture, I also talk about some lysosomal diseases, right? Okay, so lysosomal diseases. Okay, so there is, I think I talk about like Pompe disease, okay, as well as like Tay-Sachs disease. Did I? I think I did, but the recordings were made like at the beginning of the pandemic, so I haven't watched. I don't like watching myself. It's kind of weird, so I don't remember. Okay, but thank you for confirming. I did talk about it. Okay, so just quickly recap for those of you who forgot or who haven't watched the recording. Pompe disease is basically, well, let's talk about lysosomal disease in general. Because the lysosome contains many enzymes, right? And as I told you, the enzymes are proteins, and so they are encoded in your DNA. So if there is some mutation in the DNA that causes one of these enzymes to be misfolded, for example, right? We talk about the key and log relationship, right, between an enzyme and its substrate. So if you mutate the DNA and the enzyme is different shape, then they will not work. Okay, so if one of these lysosomal enzyme doesn't work, then you might not be able to break down whatever you're supposed to break down. So in Pompe disease, you have a mutation in an enzyme in the lysosome that is supposed to break down carbohydrates. So this leads to toxic accumulation of glycogen, right? Especially in the heart cells. Right? So in the heart cells, if you have too much glycogen, actually is not a good thing. Okay, it's going to impede with its function, and you know, it's could be life-threatening if it happens early on, and you know, treatment is not intervened. And then there's Tay-Sachs disease. This one is accumulation of lipids. Okay, so this one, the enzyme that was supposed to break down lipids can't be broken down anymore. So it accumulates, especially in the brain cells, and that could lead to developmental issues, and it could be lethal as well. Now, because it's a genetic disease, Tay-Sachs disease can actually be detected using amniocentesis. Does anybody know what amniocentesis is? Anyone want to explain to me what that is? Any mothers here? You would probably have heard of amniocentesis. Maybe I'll spell it for you. Maybe it's hard to hear what I'm saying. Amniocentesis. Yes, thank you, Megan. Taking liquid from the amniotic sac. That's right. Okay. So what is the amniotic sac? Okay. For those of you who don't know, when a person is pregnant, then the baby grows in the uterus, and the baby is going to be surrounded by all these fluid. You might have heard the phrase, "the water is broken." Right? That usually happens when the mother goes into labor, right, about to give birth. Then the water breaks. So the water that comes out, that's actually amniotic fluid. Someone asked me before, it's kind of fun, like, how do you know the water is going to break? And then I look at them, it's like, trust me, you would know. Because both of my time, I remember when my wife's water broke, it was like, yeah, you would know. It really comes out. Sometimes it's not as dramatic. Sometimes it kind of seeps out a little bit. But generally, when it breaks, like, it actually does come out quite, a lot of it does come out. So that fluid is called the amniotic fluid. And that's what, you know, kind of keep the baby buoyant inside the mother. And, you know, it's going to contain some baby cells. So amniocentesis involves taking a needle that it's guided by ultrasound. And then you take a bit of that fluid and then you extract the cells from it. And then you would analyze the genetic material from the baby. And based on what you see from the test results, you can detect a list of genetic conditions. Okay, so things like, you know, trisomy 21, that's relatively easy to see because you would have an extra chromosome. Other times it's not as easy to see visually, then you would have to use other techniques like PCR to check for them. And we will learn all about these in, I think, lecture 9 when we do biotechnology. Okay, so that is that. Now, let's take a quick break, like 5 minutes maybe, and we'll do more when we come back. Okay, if you have any questions during this time, please feel free to type in the check box, but otherwise go unwind a little bit and we will resume shortly. 1:35, okay, 1:35. Let's continue. Next question. List 3 organelles or structure that are unique in plant cells and briefly describe their function. So, you know, I should add something to this question. In something unique in plant cells, I would say, like, compared to animal cells. Animal cells. And I'll explain what that means in a bit. Okay. But can anyone help me here? What are 3 organelles or structure that are found in plant cells but not animal cells? That's basically what the question is now. You can just list them out in the chat. Yep, there's the cell wall, very good. Central vacuole, yeah, that's another one. Let's put it here. Central vacuole. And I'm looking for one more. Anybody? Yeah, Cynthia said chlorophyll. Chlorophyll is it's found in chlorophyll is a substance, right? Yes, and it is found in the chloroplast. That's right. Okay, so why do I have to add this compared to animal cells? I realize when I say unique to plant cells, that implies it is not found in any other cell type, but that's not true because, for example, we will find cell wall in bacteria, for example. We will find chloroplasts in some protists. Okay, so they're not exclusive to plant cell, but when comparing plant cells to animal cells, these are things that will only be found in animal cells, but not plant cells. Sorry, only found in plant cells, but not animal cells. Okay, so cell wall, what does the cell wall do? And, describe its structure and stuff like that. So cell wall is going to be made up of cellulose, right? We learned about cellulose last time, right? Cellulose is a polysaccharide, right? And it provides structural support, right? Prevent the cell from bursting. Okay. Plant cells, they love water, right? And all those water are going to go into the plant cell. And the cell wall would make sure that it doesn't blow up. And the easiest way to visualize this is think of a balloon. You blow air into a balloon. If you blow too much, it's gonna pop. If you put the balloon in a glass and then you blow it, then it's not gonna pop as easily, right? Chloroplast. Chloroplast contains chlorophyll. And why do we need chlorophyll? It is needed for, yeah, it gives color. That's correct. It makes things green. Yeah. And it is needed for photosynthesis, which is the process that converts carbon dioxide and water into sugar and oxygen. Okay, so that's how plant makes their own food. Any part that is green is capable of doing photosynthesis, and is fall time now, so you will see the leaves start to change color, right? Because there's not a lot of sunlight in the winter, so the plant needs to conserve energy. So it's gonna kind of break down its chlorophyll. They don't really need the chlorophyll anymore, and some of the other colors start to show up. Okay, so that is chloroplast. Finally, there's a central vacuole. Someone asked a question, Mikhail. There was a trend of people drinking chlorophyll. Is that bad for human digestion? I mean, it's I wouldn't say it's bad, but like, here is the funny thing. Like, if you drink any kind of vegetable stuff, like I don't know if this is gonna gross you guys out, but like when I drink my smoothies, I like to add spinach to it. Like, I don't know why, I just do. Okay, and then when I drink that, I'm drinking chlorophyll, right? Okay, and in fact, when you eat any vegetables, green ones, they're gonna have chlorophyll in it. So I don't think it's like, oh, your mom does that too, Christian. Excellent. You should tell your mom your teacher does that. It's a spinach thing. It's the iron, right? That's why I do it for the iron. But anyways, my point is, that's actually really funny. The point is, it's okay. It's naturally occurring things. Now, when you say people deliberately drink chlorophyll, can you explain? I actually never heard of people doing that. But like, do you mean like they buy? Is there like pure chlorophyll that you can buy out there? like, do they extract it themselves? Oh, really? Oh, I have no idea that you can extract. You can buy chlorophyll. I'm gonna look into that because that's interesting. But I actually do this in at school when we're doing face to face. I actually extract the chlorophyll from something like spinach, or sometimes I do it from kale, depending on what's available. And actually, chlorophyll, it's not very water soluble. Okay, it doesn't dissolve in water well. And for me to actually extract it at school, I have to add alcohol to it. And the alcohol would dissolve all the chlorophyll. And then I would have a flask of chlorophyll to show people. So that's why I asked, right? Like, I don't know like how people extract it. Do they extract it with like alcohol? If that's the case, then it's probably not too healthy, right? But if it's commercially available, I don't think it's bad. Like, it's just a naturally occurring thing. It depends on what else is in it, right? I can't imagine chlorophyll tasting too well, so they probably mix in like sugar and stuff in it as well. So yeah, I don't know. I'm gonna look that up, okay? And if I find anything interesting, I'll post it on the page. It might be one of those whole food idea, right? Like but it's just much easier to just eat your greens. No hassle. Chuck down a broccoli. You probably will get just as much chlorophyll from that. Anyways, good question. Next, central vacuoles. What does that do? Well, it stores water and nutrients. Okay. And sometimes, right, it might can also store pigments. Right? Pigments are like color, right? So like flowers, stuff like that. Or even poison. Okay. So some plants are poisonous, as you know. Where do they store the toxin? In these special central vacuoles. Okay, so those are three little things that are found in plants but not animal cells. Moving on. Okay, so now we have a bunch of other organelles, and these are not part of the endomembrane system anymore. We have the nucleus here. Okay, and I will use the red for the description. Okay, so lots of stuff for the nucleus to describe. Okay, let me zoom in a little bit here. Okay, number one, it is found only in eukaryotic cells, right? As you know already, found in eukaryotic cells. Okay, or eukaryotes, that's another way to say it. Surrounded by the nuclear envelope. Okay. And then on the nuclear envelope, there are a lot of pores. Okay. Many nuclear pores on the nuclear envelope. Okay, so this allows things to go in and come out, and I'll tell you what goes in, what comes out in a bit. Contains contains nucleolus, okay, which is this thing here in the center. So the whole thing is nucleus, and in the middle of the nucleus is the nucleolus. And also, I have to find my color back, sorry. And chromatin. Okay, so that's all these stuff here. Right, so a lot to unpack there. Where should I begin? Okay, so let's talk about chromatin. Right? What is chromatin? Well, to understand what chromatin is, and some people say chromatin, same thing. Okay, so to understand what that is, let's talk about something that you are probably more familiar with, which is chromosome. Okay. I assume you've seen chromosomes, right? On TV, whatever, on the internet and books and where else. Okay, so this is chromosome. And it typically shows you in an X. So what is chromosome? Chromosome is basically very condensed DNA fibers. It is easy to move around in the cell. Okay, and you will only usually see chromosome before, like right before and during cell division. Okay, so think of this as very packed DNA. Okay, only find chromosomes when the cell is ready to divide, because then you actually have to move the chromosomes around in the cell, right, during cell division. So DNA fiber, think of like sewing thread, right? Like sewing thread, right? Okay, it's very fine. You can't really see it too well if it's just one thread. So that's like DNA on its own. But then when you crumble it together, right, into like a ball, right, then you can see it from far away, right? So that's basically what chromosome is, right? So normally when the cell is not dividing, you cannot really see the DNA fibers, okay? But once they condense into chromosome, you can see it much easier. So what do they look like when the cell is not dividing? Well, you've guessed it. When it's not dividing, you have them in the form of chromatin. Okay, so this is basically DNA plus some packaging protein. You still have to package it, but just not to the same level of the chromosome. Okay, so packaging protein. Okay, they're called histones. You might have heard of that before. Okay. So this is less condensed. Okay, where do you find these less condensed DNA? Inside the nucleus. Okay. Nucleolus. What does a nucleolus do? Nucleolus makes ribosome. Okay, so when you finish making the ribosome, they will exit through the nuclear pores into the cytoplasm. Okay. Okay, another thing that we should talk about is, and again, I think I mentioned this in the lecture. If not, you will have to know this. Central dogma of biology. Okay. Dogma is just like a weird word. It means a theme. Okay, so the central theme of biology is that you start at information, you start at a DNA level, and then you can make a copy of the DNA in the form of RNA. And you know the difference between the two we talked about last time. And then you're going to use the RNA to make proteins. Okay. So DNA to RNA, when you make a copy of DNA in the form of RNA, we call that transcription. Okay, and this happens inside the nucleus. Once you finish making the RNA will exit through the nuclear pores into the cytoplasm. In the cytoplasm, the RNA, which has the instruction to make the proteins now, will be decoded by the ribosome, the next thing that we're going to talk about. And in the process of translation, you will make proteins. Okay, so this happens in the cytoplasm. This happens in the nucleus. Okay, so why am I talking about this? Well, I'm just telling you what comes out of the nucleus. Okay, what are all the pores for, right? So two things come out from the nucleus. There are the ribosomes, which is created in the nucleolus, and then there are the RNA, which is a copy of the DNA that you've created. And once this goes into the cytoplasm, right, we're going to use the ribosomes to decode it. Okay. All right. Couple things for the ribosomes. First of all, some are freely floating. Okay, some are freely floating in the cytoplasm. Okay. And as you know, some are attached to the rough ER. Okay. Actually, it is made up of RNA and proteins. Okay. So the actual ribosome themselves is made up of RNA and proteins, even though its job is also to make proteins, right? To decode the RNA and make proteins, right? So the ribosome itself is made up of these two things as well. Okay. So, you know, you would have the, if I zoom in a lot, then the ribosome looks like a hamburger. Okay. There is a large subunit, and then there is a small subunit. Okay, and then the hamburger, the buns, sort of, right, would be separate normally, but when it's working, right, when this is the RNA, right, it would clamp onto the RNA, and then it will read the instruction on the RNA, and then it will use that to make the protein, okay, the polypeptide. So that is the function of the ribosome. Yeah, the function is protein synthesis. Okay, and again, sometimes you can just make protein directly in the cytoplasm, and then the protein would just be floating around afterwards. Or you can make it directly into the rough ER, in which case they will be packaged and you know, either sent off or they will go to the Goji after, so on and so forth. Okay. All right, let's do another poll. Let's do another poll. Okay. So, kind of B and C are the top choices here. Let's take a look. So if you track the movement of protein produced in the rough ER, it can end up in all of the following except for, for people who chose B, that actually is not the correct answer, because the protein could end up in the lysosome, right? After all, the lysosome contains a mixture of enzymes, and all these enzymes come from ribosome production, right? Okay, and some of it could be from the rough ER. What it cannot end up in would be the smooth ER, because the smooth ER is where you make lipids, right? So the proteins that you produce from the rough ER has no business going to the smooth ER. Okay, so that is the correct answer. Okay. So let's read the question again, right? Like maybe there's just a misunderstanding of the question. It's basically asking you the proteins from the rough ER. Where will it not end up, right? So it could end up in all these places, but not, right? Except a smooth ER. Okay, so I hope that makes sense. Let's move on. Next one, we have the mighty mitochondria. That is responsible for, sorry, can write and talk at the same time. That is responsible for making energy, okay? And in terms of the structure, we already talked about it, so I'm not going to repeat, okay? So see diagram above, okay? I think it's page 1 or something like that. And the function is to produce ATP through the process of cellular respiration. C6H12O6, that's glucose plus 6 oxygen, producing 6 carbon dioxide and 6 water. And more importantly, up to 36 ATP. Okay. We will learn about this in a lot more detail in lecture 5. Okay, but that's it for now. Next, we have these French fries looking thing, or like pasta, depending on your interpretation. Centrioles. Okay, centrioles. What are the centrioles for? Well, they usually, there are one pair, right? Exist as a pair in cell. Okay, plant cells don't have it, only animal cells do. And are arranged perpendicularly, 90 degrees. Okay. What does it do? Well, it sends out spindle fibers to guide chromosome movements. During cell division or mitosis. Okay. Next, we have cilia. Okay. So cilia, in terms of description, they usually cover the entire cell. Right? What do we use it for? Cell movements or to move things around the cell. Okay, so I'll give you 2 examples. Someone talked about paramecium before. Right? So paramecium looks like this, right? There's the mouth, right? There's some vacuole here. But what's important is they are covered with cilia. Okay. So when these are protists, they live in ponds. So if they want to move around, they need to beat their cilia. Okay. So that creates cell movement, right? By beating the cilia. Okay. So cilia usually just go back and forth like this. And when they all beat together in unison, it's like, you know, a dragon bow or something, right? Everybody row at the same time. All the cilia beat in the same direction, and then they will move. Okay. In humans, can someone tell me where we find cilia in the human body? there are two common locations for finding cilia. Anybody know? Did I tell you in the class? Maybe you've read about it. Where do we find cilia in the body? No? One example is the airway. Okay, the airway. Right, so your windpipe is called the trachea, right? Okay, trachea, windpipe, and then it has lots of cilia on the inside. So if there are like dust particles, if there are like bacteria, then the cilia will sweep them up to your throat, and then you can, spit them out or swallow it. Not quite like not like within the lungs. You don't have any cilia within the lungs, but it's mostly along the airway. So trachea, the trachea comes down and it splits into two things called like bronchi, right? Yeah, the upper respiratory tract, right? There would be the cilia. And again, we'll learn it, next semester. Another one. Anybody have another one? I will give you a hint. This other example is only found in female and not males. Where else would you find cilia? In the female body, but not the male. Anybody? Ovaries close. It's close to the ovary, very close to the ovary. What's connected to the ovaries? Anybody know? The ovary makes the egg. Where does the egg go? Before the uterus. That's right. Fallopian tube. Okay, so there is the ovary on each side, and then there is going to be the fallopian tube. Okay, and then that's where the uterus is going to be. Okay, so uterus. I mean, you don't have to copy this. I'm just telling you so that it's relevant, right? Like you might be wondering, why are we learning about cilia? Why do we care? Well, it's in your body, right? So like these are the ovaries, right? Two, one on each side. And then this is the fallopian tube. Fallopian tube, or the oviduct, sometimes called. And inside, right? On the inside, there are the cilia. So the egg comes out, right? How does the egg know where to go? The egg has no cilia. It doesn't know how to move around. So the cilia inside the fallopian tube will, guide the eggs so that it will go towards the uterus. Okay, so it either moves the cell like the paramecium, or it moves things around the cell, such as the bacteria or dust particle or the eggs in the fallopian tube. Okay, this one is flagellum. Flagellum. Okay. So usually just one or two per cell. Okay, so for sperm, it's just one. If a sperm has two tails, it doesn't swim forward, it just like goes around in circle. Like you can search up on YouTube. Like it's amazing what you can find on YouTube. You can find all sorts of stuff. If you search up like, sperm with two tails, you're going to find a video, and then you will see it's just looping around in circle, right? So that one will never make it to the egg, right? It's just probably like a mitotic division error. Meiotic rather, it's meiosis. But usually there's just one. And sometimes some organisms have two, right? There is another protist. It's called euglena. Okay, EUGLENA or something like that, I think, right? And it's kind of part animal, part plant, and it has two flagella, right? What does it do? What does it do? Well, cell movement. So cilia goes back and forth like this, right? Flagellum, it's more like wave-like flapping, okay? And then it propels the sperm forward. Any question? Everybody still with me? There's no way to tell. Maybe you're not even at your computer. I would never know. I think you guys are. You guys are a good student. Okay, thank you. Thank you for confirming in the chat. Moving on. This is pretty straightforward. If you need help memorizing the parts, maybe change this into cue cards, and you would definitely be able to, memorize it after a few flips. So let's go through them. Directly involved in protein synthesis. That would be ribosomes. Okay. The region inside the cell, except for the nucleus, is the cytoplasm. That's where everything else is submerged in, right? Bathed in. Organelle that manages or controls all the cell functions in a eukaryotic cell. That's the nucleus. Digest excess or worn out cell part, food particle, and invading viruses or bacteria. That is the lysosome, part of the endomembrane system. Next, small bumps located on portions of endoplasmic reticulum. That is the ribosomes again, making it rough ER. Involved in detoxifying harmful substances and digestion of fat, produces hydrogen peroxide as a byproduct. At first glance, you might think this is talking about lysosome, right? And it's very similar to lysosome, but the giveaway thing here is hydrogen peroxide as a byproduct, okay? So that tells you it is actually peroxisome, okay? So peroxisome, it uses oxidation reaction to break things down. Okay, oxidation reaction means you strip electrons from something. That's what oxidation means. You rip electrons from them and break it down. And the byproduct is hydrogen peroxide. Hydrogen peroxide is H2O2. It's very, very toxic for your cell. That's why all these reactions must happen in the peroxisome, and then the H2O2 will get broken down into harmless water and oxygen. Okay. If this H2O2 gets out from the peroxisome, cell's gonna die. We don't want that. Produces a usable form of energy, that is the mitochondria. Sorts and modifies contents of vesicles, Goji Body. Site where ribosomes are produced, that would be the nucleolus. If you think about like a peach, the skin would be the nuclear envelope, right? The meat would be where the chromatins are found, and then the seed inside the peach, that would be analogous to the nucleolus. Provides support for the cell. This is a cytoskeleton. There are 3 types of cytoskeleton that we talked about. I think it's at the end of the lecture. So there are like micro filament. Okay, and it's made up of something called like actin, right? So this is used for cell movement. You have a lot of actins in the muscle. Muscle is basically made up of 2 proteins. One is actin, the microfilament. The other is myosin. They grab onto each other, and that's how you contract muscle. The second one is the intermediate. Intermediate filament. And these are not for cell movement, but rather they provide structural support, structural stability. They help 2 cells to join together, for example, forming these so-called tight junctions. Okay, so for more information, read the PowerPoint slides, watch the recording. And the last one is the microtubules. Okay, so in the cell, if you think about it, like this is the Goji body. Sorry, this is the nucleus. This is the endoplasmic reticulum. This is the Goji body. You don't have to draw this, just chicken scratch, okay? So when a vesicle comes out from the rough ER, like how does it know where to go to get to the Goji body, right? Okay, like it doesn't have legs or anything. Like if we rely on diffusion and chance, it might never get there. So it turns out there are these microtubule tracts all over the cell, okay? And then this would hop on to the microtubules, and then it would transport along it to get to the Goji body, okay? I am not making this up. This is like, again, you can watch it on YouTube, but this is true. It's so cool that there is a microtubule, okay? And then there's actually a molecule that looks like a little man with no head though, a headless man, okay? There's a protein like that, okay? And it literally carries the vesicle, and then it will walk along the microtubule and take the vesicle to the Goji body. Like they've showed this, like in real life, you can see the legs detaching and attaching, okay? It is super cool if you ask me, okay? So go search that up. Anyways, I went on a tangent again, didn't I? Next, small hair-like structure used for movement. That is a cilia. Cilia. Forms a selective permeable membrane around the cell. Cell membrane. Long whip-like structure used for movement. Flagellum. Contains chlorophyll, needed for photosynthesis, that is chloroplast. Large sac filled with water in plant. Central, vacuole. Produces microtubules, fibers needed for guiding chromosomes during cell division. Centrioles. Right, so here is the microtubules is the spindle fibers, right? That's what they're made up of. And it serves as guiding system, just like I told you before. All right, long answer question. Only 3 long answers this time, and I think they're pretty self-explanatory. But does anybody want me to take up any of these, 1, 2, or 3? If not, I will just do the application question. So we're kind of running tight on time anyways. Anybody have any requests? Okay. I mean, number 3. Okay, yeah, sure. Compare the structure of mitochondria and chloroplast. So we did mitochondria already, right? Like above. Maybe I'll just quickly go through the chloroplast. I don't know if I did the chloroplast in the lecture. Anyway, it doesn't matter. So here it is, right? So this is the chloroplast. Just like mitochondria, the chloroplast is also double membrane. So it has an outer membrane. Okay, so mitochondria has an inner membrane like this, right? You know, like this, right? Some people say it's like an accordion, right? Okay, so this is the mitochondria, but for the chloroplast, the inner membrane kind of form these, you know, stack of, you know, I'm trying to think of analogy, like, you know, like those jelly-filled donuts, if you know what I mean. Are they actually called jelly? I think they are. Like you know, the Tim Horton ones, right? That comes with like white icing on it, and then inside is filled with like red jellies, right? Or like yellow jellies, right? So it's like having a bunch of those things stacked together. I don't know. That's how I imagine them. Okay, so that would be the inner membrane, and that each of these things, this whole thing, is called a thylakoid. Okay, and the membrane, the inner membrane, right? Instead of forming the cristae like we saw in the mitochondria, these would be called the thylakoid membranes. Okay, the inner membranes. Okay, that surrounds the thylakoid, and inside the thylakoid, if you like my donut analogy, the jellies, okay, the inside, that's the stroma. Okay, so the stroma is analogous to like the matrix, I guess. Oh, sorry. I'm sorry. I made a mistake. The stroma is actually the fluid outside. So not the jelly. Okay, I got too carried away with the analogy. Stroma is the fluid inside, and then the inside of the thylakoid, you would have the thylakoid lumen. okay, the jelly inside. So that would be like the matrix. Okay, so it's similar in the sense that they both have membranes inside a membrane, and then there are fluids separating the membranes, right? So the stroma is more like the intermembrane space, I guess. Okay, and just like the mitochondria, chloroplast also has its own DNA, right, which helps make some of its proteins. Okay, so mitochondria is where cellular respiration takes place, and chloroplast is where photosynthesis takes place. Okay, so the chlorophylls are actually found on the membrane of the thylakoid. Okay, I don't think I asked you many questions about the chloroplast structure in the test. The focus is on the animal cells. I hope that answers your question. If you have follow-up, please let me know. Okay, let's talk about these application questions. First one, speculate what will happen to an animal cell if it is placed in pure water. So we sort of mentioned this earlier, right? If you have water here and then you put an animal cell in it, because animal cells have a lot of stuff in it, right? Like the organelles, the nutrients and stuff like that. What's going to happen is the water will try to go in, and it will try to dilute the contents of the cell. And what ends up happening is that the animal cell will swell and burst. Okay, so did you know drinking too much water can actually kill you because it dilutes your blood, and then your cell will swell up. And if you drink too much, then the cell will swell up and burst, and that could kill you. It has happened before, and I think I mentioned it in the next lecture recording, so I'm not going to spoil it for you. You can watch it and find out what happens. Now, if you do an animal cell instead, if you do a plant cell instead, the plant cell will have a cell wall, right? So even though a lot of water goes in, is not going to burst. The cell wall will protect the cell from bursting. Okay, so if you water your plants regularly, they will stand up, right? And if you stop watering it, then it will start to go down and wither. Okay, so plant, they love to have all these water going in them. They would just store them in the central vacuole, and then they will help the plant kind of perk up and stand up strong. Okay, next one. Your heart muscle needs to function continuously throughout life. Which organelle would you expect to be most abundant in heart cells? That would be the mitochondria, right? Which makes sense because mitochondria makes ATP, and you would need lots of ATP for something that keeps on beating. Okay. Now, last one is the one that I want to see some answers from you guys. Endocrine glands produce and secretes hormones. Which organelle would you expect to be most abundant in the cell of an endocrine gland? Justify your reason. Okay, so go, tell me, what do you think? What organelles would be most abundant and why? Okay, so Safi said ribosomes. Yeah, there is going to be that. Anything else? Anyone else? Anything else? Like, what if I make this into a multi-select question on the test? Select all the organelles that are expected to be abundant in an endocrine cell. So besides ribosomes, what else are you going to choose? Are you going to choose anything else? Anyone else? Don't be shy. Smooth ER, says Megan. Yeah, that's also correct. All right, so I'll just tell you the model answer. This is a trick question because it depends, right? Hormones. What are hormones? Can someone remind me what hormones are? Like, what are they made up of? Please tell me. Yeah, proteins. Are all of them proteins? Are all hormones proteins? Steroids. That's right. And steroids are lipids, right? So if the endocrine gland makes protein hormones, then you would expect to have rough ER, right? Lots of rough ER, ribosomes, and Goji. Right? If it makes steroid hormones, on the other hand, you would expect to have smooth ER instead, right? That's where lipids are being produced. Okay, so it's kind of like a trick question. That's why it's application question. But I hope it makes sense, right? Any question before we do one last poll and say goodbye for the week? Okay, poll it is. Last poll. I will try to, like some of you, the survey, right, that I told you guys to do. If you haven't done the survey, please do survey. Give me some feedback. And some people say they want more of these poll questions. So I'll try to make more for future lessons. But here is another one. Oh, sorry. This is a multi-select question. I launched the wrong poll. Let's try again. Okay, let's see. False. False statements. First one, smooth ER is part of the endomembrane system. We know that is true, right? The endomembrane system contains the rough, the smooth ER, the Goji body, as well as the lysosome. So that is a correct statement. It's not the answer. Muscle cells are expected to have an abundant mitochondria. We just talked about that in the application question, so we know that is true. There are multiple nuclei in single brain cell because high processing power is needed. That sounds tempting, but cells would only have one nucleus, so that is not correct, and that would be that's a false statement. So that's something you would circle for this question because we're looking for the false statements, right? All cells have cell walls to protect itself from the environment. That is absolutely not true. Animal cells do not have cell walls. Okay, so that is that. All right, so what's going on next week? Well, let's take a look at the calendar. For next week, which is week number 4, if you can believe it, you will do your regular asynchronous stuff, right? Watch the recordings online, and then we would have a tutorial again. Oh, this is my other class, but anyway, for you guys, it would be like 12:30 in the afternoon, right? Okay, and then the following week, there would be no tutorial for week number 5. You will just be doing your beetroot labs. Okay. In fact, I encourage you to start working on it, and then you can hand it in like early rather than later, because there's also quiz number 2 coming up. Okay. So I will actually post a link on the debrief this week to tell you where the lab is in case you cannot find it. Okay. And then after that, you will have the engagement week. So well, I assume you guys, this is like first semester in the college ever, right? So an engagement week, it's kind of like reading week. There's no school. College offers some, workshops and things for you to join, but those are optional. And it's basically a week to, let you take a break. And there are no assignments due on engagement week. And for my course, there are no quizzes or tests afterwards as well. So you can just, forget about bio a little bit, but don't forget about it too much, because then the test is at the end of October, and that is the entire first half of the course, lecture 1 all the way to lecture 5. So yeah, that's that. If you have questions, need help, whatever, right, just contact me and email me. I'll get back to you. Otherwise, have a good weekend, and I will see you all next week. Bye for now.